Now, please, Dr. Matus, you can start. I can hear you.
Yes. Okay, now it's okay. Good morning, everybody. I would like to welcome you to all participants to this webinar titled Carbon Cycling in Maritime Antarctic Soil, Impact of Soil Carbon Sequestration. This webinar is organized by Laboratory of Conservation and Dynamic Volcanic Soil, and the two running projects support. The first one is the INACH project, which is a Chilean Institute, Antarctic Institute, and a Network Extreme Environment project, which is called NEXER. Today, our webinar is going to be moderated by Mrs. Maria Javiera Guarda. Nowadays, she is part of BioREN and NEXER project outreach team, and she's going to moderate our talk. Thank you very much for your participation, and Javiera.
Thanks for the wonderful introduction, Dr. Matus. As he said, my name is Maria Javiera Guarda, and today I will be your webinar host. The following talks will last half an hour approximately, and later Dr. Matus will host an instance for questions at the end of both presentations, so our audience can have the freedom to ask our expert guest until 1 p.m. official end time of this webinar. I would like to ask the audience to silence your microphones during all the presentations. So, first, I will introduce Dr. Kelly Wilhelm and later Dr. Francisco Matus. The questions during the presentation are going to be through chat or later raising your hand, as I said, at the end of the two talks. So, Dr. Kelly Wilhelm is assistant professor from the University of Wisconsin River Falls from 2019, giving lectures in soil conservation and natural resources related topics. He has more than 10 years research experience in soil science and related topics. He joined INACH project from Dr. Francisco Matus to apply in 2019 to Fulbright Scholar, which was awarded in 2020. Dr. Wilhelm's presentation is entitled Soil Development and Active Layer Dynamics, Climate Change on the Antarctic Peninsula. Dr. Wilhelm, the audience is all yours.
Thank you very much. I'm going to share my screen so I can have some images along with what I'm talking about. Can you see that okay?
Yes, everything's okay.
Yes.
Okay, so like I said, my name is Dr. Kelly Wilhelm, and I am an assistant professor for the University of Wisconsin River Falls. And I'd just like to thank everybody for attending, and thank you to the Chilean Antarctic Institute and the U.S. Fulbright program for the opportunity to give this presentation and do some research alongside them. So my presentation here is going to be just kind of a summary about my background and some of the research that I have done that is related to the work that I'm planning to do in Antarctica. So just a little bit about my background. Maria gave a very good summary of it. A couple of other things
Sorry to interrupt you. The presentation is not very good, not very shaped in the screen. Can you fix it a little bit?
Let's see.
Now, yes.
But, you can see that okay?
Yeah, you have to put in the presentation screen mode.
Okay, so if you can see it okay, we'll continue from here. A little bit more about my background. I got my undergraduate degree from the University of Iowa in geoscience and archaeology. And during that time, I used mass spectrometry to try to relate lead isotope ratios and find where archaeological artifacts actually came from. I continued on with my education at the University of Wisconsin-Madison, where I started looking at soil science and really focusing on nutrient cycling in forested areas. And for my PhD, I focused on soil physics and temperature propagation through Antarctic soils. I worked as a conservationist for the US government for a couple of years before moving on to work for the University of Wisconsin, Green Bay and River Falls as a lecturer and assistant professor where I currently preside. And so I'm going to talk a little bit about what I did primarily during my time at the University of Madison and hopefully be able to talk a little bit about what I'm planning to do in the future.
Sorry, can you put in mode, screen mode, your presentation? Yes, thank you. Oh.
Yeah, so, does it become fuzzy if I do that?
Yeah, well, yes, go back, please.
Oops.
Okay, sorry.
So I can maybe try sharing it in a different screen. Let's go here. Does that look any better?
Yes, it's good. Now
okay.
It's much better.
All right, so my PhD work focused on the Antarctic Peninsula down near South America. It's not a place that a lot of US researchers go down to look at soils. A lot of research is focused around the McMurdo area, just because it is a larger base. We have the dry valleys in that area. But for the most part, I chose to work in this area because it is an area that is experiencing a lot of changes in recent time, temperature-wise and climatically speaking. In this area, I focused on two different locations, Amsler Island, which is where the US facility is located, and on Cierva Point, which is an Argentinian base just to the north. And these areas were chosen, and the upper area of the peninsula itself was chosen because of the extreme warming that it's experienced. It's been isolated as one of three major areas experiencing the highest temperature changes on the planet, alongside Siberia and Alaska in the northern hemisphere. So this is an area of increasing interest in the effects of the warming climate and the changing climate dynamics in this area. And not very much research has been done on the soils of this area that I wanted to be able to add to our knowledge base there. And just some information about this area: between the 1950s and 2017, temperatures increased by over 3 degrees Celsius, which is approximately 6 times greater than the planet on average. And it's predicted to continue at this rate for the foreseeable future. And so better understanding of soil development in this area and in carbon sequestration is going to become of more and more interest as time goes on, as glaciers retreat and more land is exposed for soil development. And the peninsula is another area of interest because of the large amount of area that is already ice-free. in this region. So the Antarctic continent in general is less than 1% ice free. However, the Antarctic Peninsula is about 14% ice free, give or take on those numbers. But that means that a very large proportion of this area is ice free or becoming ice free at a very accelerated rate. And so looking at the soils of this area becomes of primary importance as temperatures increase and more interactions with the atmosphere are occurring. And the Antarctic Peninsula is a region that is of primary importance, like I said, because of the temperatures. The peninsula is mostly above the Antarctic Circle, and climatically speaking, it is unusual compared to the rest of the continent because of its warm temperatures and its large amount of precipitation that it receives every year. And so the soil development in the Antarctic Peninsula region is going to be happening at a much accelerated rate compared to a region such as the dry valleys around the McMurdo area on the opposite side of the continent because of the higher temperatures and the increased precipitation. So for my project, I looked at primarily two different features. One being the active layer dynamics, where we were looking at what is happening in the ground temperature-wise and what is controlling changes in temperature within the ground. The second thing that we focused on in this project was soil development. What is happening chemically, biologically, and physically speaking to the soils as they are exposed for longer periods of time to atmospheric conditions. And if I have time at the end of this, I'll kind of speak to what I'm looking for in the future for this project. So I'm going to start out with active layer dynamics, looking at what our project examined and found within this region. So, like I said, I'm looking at the Antarctic Peninsula and focus primarily on active layer temperature dynamics. Now, the active layer is the space between the surface and the permafrost table within a soil or a bedrock. So it's a temperature-related feature. But it is of great importance, especially in this region, because it is the region in these soils where you're going to have liquid water, chemical reactions, biologic functions happening, and vegetation growth happening. And so the understanding how deep and how long throughout the season that this active layer is going to be prominent is going to be something that we were interested in examining and being able to more actively point to. In this region, we have depths this deep, so we're having functions happening at certain depths or certain times of the year. And on the Antarctic Peninsula, for the most part, active layer temperature dynamics had been monitored pretty heavily in the King George Island area and the Livingston Island area, where there's a lot of bases and stations from countries around the world, and farther south in the Rothera area where the British have a base. However, the region that we were interested in was right in the middle of this, where there was no temperatures that had been taken underground for a very long time. Plenty of atmospheric temperatures had been taken, but very little soil examinations had been done in this area, because the US base in this area is mostly focused on marine studies and climate studies rather than terrestrial studies. So very little information was available along this stretch for temperatures dynamics that are happening underground. And the last time that this region had even been examined for soil properties, soil formation, or soil temperatures was back in the 1970s. So we had a gap of 40 years of information in this region that we just did not know what was happening in these soils. And all we could do at this point was look at Livingston and King George and Rothra and try to estimate a gradient between those two to see what the temperatures may have been in this region. But we wanted to get a better understanding of this gap. And so we focused primarily on Palmer Station, which is the US base on the Antarctic Peninsula. And this has a pretty long history of occupation. It's had residents since the 1960s. The upper image here is of the original Palmer Station. I think this picture was taken in 1965. Not much of a station back then. Today, the station is much larger, and they actually had to relocate the station from the 1960s. In the 1970s, they moved it to a different location because originally it was located right here. That's where this station is. And they thought it was part of the main island. However, it was discovered as the glacier started to retreat that this was actually just a very small sub-island. And so they moved the station from this area where we see this down to where it's indicated on the map today. And if you look at this image, I'm standing on the island where the old base used to be, looking towards the new base. You can't really see it too much, but it's back here in the background of this image. So there's been a long occupation in this area, long continuous occupation. And this region has a fairly complex glacial history. You have the Mare Ice Piedmont back here, but it's been retreating over the last 50 or so years, exposing a lot of land to the climatic variations, temperature variations, and precipitation. And this lower image is one of my previous trips down there in 2011. This valley is this valley that you see back here, just looking at the opposite. So I took the picture standing up here on top of this ridge. And now this valley is completely unoccupied by any active glaciers. They've all retreated far enough back that the only glaciers that we have are these just little ice fields that don't contribute a whole lot to the liquid water movement through these valleys. And this image that was taken in the 1970s, or this map, it's very outdated now, of course, but now the glacier has retreated much farther back here, several hundred meters of glacial retreat. Oh, one other thing I wanted to point out here is the valley that's depicted in this image has some fairly deep soils in it. We were able to dig with a shovel without any extra equipment down to about 2 meters in this region. So we can get some pretty good ideas of soil development happening in this region. And that's not to say that we find soils this deep all over throughout this region. This is a very contained valley, and so you get a lot of accumulations of soil in this valley. For the most part, soil depths in this region ranged from a few centimeters to maybe a meter in depth, but we were able to dig down almost 2 meters in some areas. But going back to the glacier movement, like I said, this area has been occupied for a fairly long period of time. We have good glacial records of the Palmer region, and glacial retreat here has been pretty significant. Around the US base, the glaciers have retreated approximately 400 meters in 50 years. So this is Palmer Station in the reds, the new Palmer Station in the red circle here. This is the extent of the glacier in 1975 and the retreats in the subsequent 50 years. This is the same image a little bit closer and rotated 90 degrees. And you can see there's been some pretty significant retreat. The base was actually built right at the base of the glacier. They could literally walk out the door and touch the glacier in about 10 seconds. Now you have to do about a 20 to 30 minute walk before you can get to the glacier. So all this land in the last 50 years was not exposed to precipitation and exposed to the atmosphere until recently. And so in this region, we have very incipient, very recently exposed land and very newly developed soils in these regions, giving us a good opportunity to look at what is happening in initial soil formations of these regions. For this part of the project, we were really focused on temperature dynamics, whether it was in soils or in bedrock. And so we installed a network of temperature sensors throughout Palmer Station and the other site that we looked at, Sierra Point. These sensors were of a wide variety. We drilled 2 deep boreholes that went down about 15 meters with temperature sensors at regular intervals. We also drilled about 30 shallow boreholes that varied in depth between 1 and 2 meters. And we also installed climate stations, like you see me installing right here, that included several soil temperature probes that were at several depths down to about 1.6 meters. Additionally, anywhere that we were able to dig soil pits, we had a handheld soil temperature probe that we could just stick in the soil to see what the temperatures were. And typically we were looking at about 1 meter depths for temperatures in these regions. One of the very unusual findings that we had in this study was that we could not find the permafrost table with any of our soil pits, nor any of our shallow boreholes. We went down to 2 meters and we could not find where the temperatures were at 0 degrees Celsius or below. And so that in itself is a little bit unusual because we're farther south than King George Island or Livingston Island. And I'll go back to the original image here. These numbers here were the depths that they would find the permafrost table at. And so for the most part, they were finding it between 1 and 2 meters. There was an outlier at 4 meters, but for the most part, they were finding about 1 meter depth before they hit the permafrost. Down in Rothra, they were finding approximately the same thing, about 1 meter before they hit the permafrost table. However, us being in the middle, we expected to be about 1 to 2 meters. But our results and our data were telling us that we weren't even close to the permafrost table. This is a temperature profile from our climate station. So just one of the methods that we used to monitor temperature in the soils themselves. And we compared that to air temperatures, surface temperatures, and temperatures down to 1.6 meters. And right here is our 0 line where we would, if we were to find temperatures that remain below 0, that's where our permafrost table would be. But as you can see, even at our deepest depth, during the warm season, temperatures were well above 0 degrees, up to 5 degrees Celsius for the most part during the warmest period, which is indicating to us that even our deepest depths that we could reach with a shovel or a shallow drill was not getting us even close. And to us, that was a little bit unusual because we were expecting to be getting pretty close to the permafrost table based on what we were seeing in other regions. Additionally, the last time that somebody took temperatures in this region, they found the permafrost at about 30 to 50 centimeters. And we weren't finding anything close to that here. Well, we couldn't detect or actively measure the temperatures with these climate station probes or with our shallow drills. So we had to use some other methods to try to determine where or how thick our active layers were or how deep it was until we got to the permafrost table. And what we found was using equations and using different modeling programs, we found that we would estimate that the active layer was going to be as thick as 9 meters in these soils, assuming that the soils were continuous, which is a bit of assumption. But even in bedrock, we were finding our calculations to tell us that temperatures were going to be fairly warm, very deep in these soils. And so that was an extremely, extremely unusual finding. These calculations telling us that active layers were almost an order of magnitude deeper than regions farther to the north where it should be warmer. And we used models to try to see if we could find different findings. This is a model looking at the near surface temperatures, comparing both measured data to modeling data, and you can see they match up fairly well, giving us confidence that our models are going to be fairly accurate. Well, these models found that this very similar thing to the equations that we use, that we weren't going to find permafrost until we were getting down to nearly 10 meters. And if equations and models weren't enough, we did have those extremely deep boreholes that did have temperature sensors in them. And these regions, we did find the permafrost. And we found it exactly where our models and our equations said that we should, about 13 meters deep, 13 to 14 meters down here. So very unusual findings here, not something that's expected given the information that we had from surrounding regions. We looked at similar temperatures and did a similar study at Sierra Point, which is a little bit farther to the north. Down here was where Palmer was. Sierra Point is a bit farther to the north, so we expected it to be a little bit warmer. Couple of different features from Sierra Point. It's more sloped. Oops, you can't see that image, but that's okay. It's a bit more sloped than what we had in Palmer Station. Not as deep of soils, and we had the added influence of a very large penguin rookery and large amounts of mosses growing in this region. And we looked at the temperatures of this area and found that, as we expected, the active layers were thinner here. Or sorry, not as expected, but unusually so, the active layers were thinner farther to the north where it should be warmer. Here we found active layers to be about 7 meters deep compared to 13 farther south. And so we were at a bit of a crossroads of how to explain this and be able to put it in the context to compare to King George and Livingston Islands and Rothera farther to the south. Because if we had found something like 2 or 3 meters, that's a pretty reasonable offset from those more northern areas. But to have 7 to 13 meters, that means that something's happening here to affect our active layer depths. And so we tried to correlate these depths to all kinds of different features, from climatic features to local topography and different geologic features, hydrologic features, whether or not there's organisms or all kinds of different things to try to figure this out. Climatically speaking, there was nothing unusual about this area. Climate has temperature, precipitation, solar radiation, all that has been monitored for 50+ years in this region. And it follows the gradation that you would expect going from a warmer northerly area to a more southerly area. And so we didn't find any good correlations between climatic features here and the unusually deep active layers. We also found that the extreme depth of these active layers meant that the superficial features, like the soil properties or any organisms living at the surface, just had a very limited effect on temperatures this deep down. If you have soils that are a meter deep, they're not going to be affecting temperatures at 9 meters. So we eliminated those as features because we were seeing this both in soils and in bedrock. So we eliminated superficial features as a primary component. Additionally, proximity to glaciers, geomorphic features on the surface, there just was no correlation between these and our deep active layers. The one thing that we did find was that the bedrock in this region had some features that were indicative of something unusual happening to create these deep active layers. The bedrock in both Palmer and Sierra were highly jointed, very large cracks in these rocks that went very, very deep. Additionally, in these areas, we had a lot of standing water. This is at the summit of the highest point of our research location. Pure bedrock in this area, but we had very large ponds of water. In fact, we used these ponds to cool our drills, and it was a very prominent feature at the top of these hills to have standing water. But it was also a prominent feature to have these joints. The shape of this pond you can see is going with the geologic features here. And so we associated this region with having very enhanced downward movement of water deep down into the bedrock or the soils. And this water is bringing heat with it, warming up the surrounding area enough that the active layer is going to be showing up as deeper than what we would have expected in a pure solid bedrock. And so that's the only thing that we could really find as having good correlation, and the only reason that we could see that our active layers are this deep compared to other regions. We think it's a factor of the highly fractured bedrock enhancing liquid water movement deeper into the soils, helping to thaw the active layer deeper. Additionally, in the soils themselves, we had a lot of active water movement, seasonally speaking, as snow and glaciers were melting every summer with increased temperatures. This is just from the bottom of the valley, and you can see a lot of fluvial river and active water movement that were depositing soils. So we have a lot of liquid water in this region that is promoting higher temperatures than what we would have expected based on other regions. So the other thing that we were looking at were soil properties and carbon sequestration in this region. Palmer Station, this is all the same information I had given before, but in this region we had very little vegetation. We had a bit of moss, maybe a bit of grasses that were developing. But we did have some seal wallows. These were very prominent in this image. This black splotch here, this is an elephant seal wallow. So we had regional areas of animal activity that actually did have effect on temperatures, but also on soil development of these areas. We had active water movement. I had shown this image before, but we had some very good distributions of soils and developing soils in this region. So we had sands and loams mostly coming from depositions from the glaciers, with a wide variety of bulk densities, so different compactions and soil particle interactions. We also had a pretty wide variety of water content throughout our soils, mostly related to the texture distribution of our soils, and a pretty large range of organic carbon contents, and I'll talk about that in a moment. But the vegetation, like I said, there's not a whole lot in this region, very thin mosses and lichens, and a couple of regions of grasses. Looking at carbon was one of the things that was a bit interesting in this region, because we wanted to see if carbon development and sequestration were related to soil properties in this region. And so we looked at soil properties and tried to do some simple correlations between carbon content and bulk, those different properties. And we found that bulk density pretty much didn't have any correlation to the carbon development, which is a bit unusual here, because with bulk density, you have different interactions with the organisms that would be helping to sequester carbon, but we weren't getting any good relationship there. We had a better relationship, still not great, but a better relationship between carbon and texture, so the percent of clay, and water content. These aren't great correlations, but they're better than what the bulk density was showing us. So we can see that the retention of water and the smaller particle sizes are helping to retain carbon for longer in these soils. Additionally, not much of a correlation between distance from the glacier and carbon, but you can see an interesting pattern forming here. You had higher carbon contents right at the margin of the glacier, and then a marked decrease in carbon. And then as you got farther away from the glacier, our carbon content started to increase once again. So we related this as when you're close to the glacier, you have a lot of water available, water being something that organisms, whether it's plants or microbes, are going to need. And as you get farther away, that water source starts to become less and less. And farthest away from the glacier, you're going to have a longer time period for carbon to start to develop. But this leaves the question of, were there other environmental features that we didn't make comparisons here? Because the main point of this study of soils was mostly just to collect data on what soils were even developing here, rather than trying to make a relationship to carbon. So we didn't have enough sites to really make good assessments, but this is some interesting preliminary data on those relationships. One thing that we did find, that geomorphic features within this region were something that was actually did have a good relation to carbon or organic matter content. So we were looking at things like moraines, stone circle formations, and mud boils, all things that you're seeing here. And in each one of these, we found that we had a markedly higher carbon content, regardless of their distance from the glacier or any of the other soil features. We found that specific geomorphic features had much higher carbon and organic matter contents than surrounding regions. So this is something that was a pretty interesting find, and something that I'd like to look into further in potential future studies, because it wasn't really the original purpose of our study, so we didn't have the data set that we needed. At Sierra Point, we were looking at soils similarly, but this is an image of Sierra Point, and you can see it's much more sloped, but it had different features to it, such as those penguin rookeries and very, very thick moss accumulations. So here are the moss and some of the penguins. These had a very marked effect on our soil developments that we weren't seeing in Palmer Station. And so that was something that we wanted to focus on in Sierra Point. Some of the mosses that were building up in this region had been there for 20 plus years, and we found in some spots that they were over a meter deep of just organic moss before you even got to bedrock or soil. So these are going to have a pretty big effect on soil development and carbon sequestration in our soils. At Sierra Point, we categorized our soils based on different properties, chemical properties, and whether or not there was moss or ornithogenic development occurring. Ornithogenic meaning related to penguins and birds. So we combine these into categories or subgroups to try to see if there was any distribution of each of these features, geographically speaking. And so you don't see a whole lot of change between some of these profiles. Visually and texturally speaking, our different soil properties, there wasn't much variation that you could see. It was a chemical difference. Profile C here, this is one of our moss. Here it goes down to 60 centimeters, but this is all organic matter. And over here you can see, don't know how good the image is for you guys, but you can see at about 17 centimeters or so that there's a change in color. And this actually correlates a lot with the change in chemical properties. Things like a lot of phosphorus coming from penguin guano developing at the surface. And so that really changes our soils, chemically speaking. So based on these three categories, we mapped out all of the soils of Sierra Point that we had examined. And these pin marks on this map were of these three categories. The yellow is looking at penguin related soils, greens are moss related, reds are acidic soils, and blues are neutral soils. We didn't find any basic soils. But what we did find here was that there was an interesting distribution between the two chemical categories, the acidic and the neutral soils. We found that the neutral soils were mostly found along the glacial margin, and the acidic soils were found much farther away. And for the most part, the reason for this is when the bedrock is originally exposed by the glacier, the decomposition or the breakdown of that bedrock is going to be pretty neutral. They are granites, and so they don't have a lot of acidity. But over time, they will start to become more acidic as you get carbonic acids and different acids being deposited atmospherically. You can note that down here, we do have an acidic soil at the very southern part of our map. So this map is looking at the northern area being the lowest region and going up elevationally as we go south here. And so this doesn't seem to match our findings everywhere else. However, this is the peak of our region. And so this area right here probably did not have glacial cover when this whole region was covered by a glacier. This was probably too high up. And so it was peaking out above our glacier. And so it has been exposed for a much longer period of time. Number 3 here, this blue one, is going to be more neutral because here we actually found that it was a dip in the geologic features. And so this was probably covered very recently and has only been recently exposed. And so the penguins and the moss areas were much farther or much closer to the water line and much farther away from the glacier. And as you get closer to the glacier, our soils became more and more neutral as they had less exposure to the atmosphere. Finally, we looked at carbon of this region, and not unexpectedly, carbon was highest where we had organisms, more organisms, where we had the mosses and where we had the penguins. But we also found that in the acidic soils that had been exposed for longer, we did have a significantly larger accumulation of carbon when compared to the neutral, recently exposed soils. And this all goes into trying to be able to assess, okay, how is carbon developing in these areas as the glaciers retreat? Because we look at areas like Alaska, and Alaska 50 to 100 years ago were just covered by glaciers. They've retreated excessively quickly, exposing large areas. These are the same exact locations. You can see the mountain in the background here and here. It's now covered by meadows, wetlands, and even forests. Now, we don't expect this to happen in Antarctica anytime soon, but with all of this vegetation growth in areas where glaciers once were, we need a better understanding of the carbon dynamics in this area. So probably took a little more time than I should have, but if you have any questions for me, please feel free to contact me.
Thank you very much, Dr. Wilhelm,
Sorry, let me
for your interesting presentation. I'll recall you for the questions at the end of both presentations. So now I would like to introduce Dr. Francisco Matus from Universidad de la Frontera, Director of the Laboratory of Conservation and Dynamic Volcanic Soils. Dr. Matus is a full professor in soil science and environmental sciences from Wageningen University from Holland. He fulfilled his career as agronomist and later he is master at Pontificia Universidad Católica de Chile. He has vast experience in soil science, but particularly in soil carbon sequestration and related topics. His presentation is entitled Carbon Dioxide Emissions Along the Chrono Consequence of Soils Development After the Glacier Retreat in Maritime, Antarctica, King George Island. Dr. Matus, the audience is all yours.
Thank you very much Javiera, for nice presentation. Thank you, Kelly, for very interesting presentation, but we have also common result that I'm going to present here. Sorry, my voice is a little bit problems now, but so we have been working in two large projects, which is called INACH and NEXA project. And the idea of this project is to look at more or less the carbon sequestration in those areas. So we have been working in Shetland Island, particularly in Fildes Peninsula. You can see here. It says farther north that the Kelly was working, but here we have a very variation of soil, a lot of variation in soil, and this is the glacial retreat from many years ago, about 200 years ago. But the Ardley Island is also a nice point because it's a small island close to Escudero Base. So, sorry, I can this, okay. Here it is. And this island is made of the soil. It's very influenced with the penguin rockeries and also birds nesting. So some general point that already Kelly showed part of it, but this is exclusive for Peninsula, Fildes Peninsula. Is about 30 square kilometers. And the region is subantarctic, cold, moist, maritime climate. The parent material mostly is lavas, a small outcrop of tough volcanic sandstone agglomerates. The mean annual temperature is very close that Kelly showed us before, minus 2.1 Celsius degrees, and in summertime about 0 Celsius. Precipitation is about 350, 500 millimeters per year. And also the Western Antarctic, already Kelly told us, warming is the fastest place on the Earth, 0.9 19 Celsius degrees per decade. Well, we have a little different figures, but it's more or less in the similar range. The ice free after the last glaciation is about 8,000, 5,000 years ago. This is the most southern part of the island. But the retreat of glacier, we have been aware that it was 200 years, more or less very close to the ice front. So the permafrost is sporadic, over 20 meters above the sea level. You might find more continuously permafrost about 30 or 40 centimeters deep. So you know that soil formation is the result of climate, of course, parent material, soil organic matter, relief time. And soil organic matter is very important point also for soil carbon sequestration, and it is determinant factor on soil formation by the interaction of soil microorganisms and minerals. So my question, or our question in the project, is how fast these processes speed up our underwarming pressure that we have been facing for the last 50 years. And this is a question that strike us, and we try to find a little bit more answer on that. This is a Scudero-based view. This is a middle island, Fildes Peninsula. We're looking for farther down there in the glacier, Colin glaciers. So this is the basis that we are going to going there in the next expeditions. So here is a very recently published map about the soil. This area, even when it's more studied from marine point of view, but there are some studies in soil, but in geology also some support information. But actually the relationship between the factors of soil formation are very scarce. You don't find just the publication told us the description of the soil, what is going on there, but there is few results about the relationship or the interaction of those factors. We have been working or sampling more than 14 soil points here, very close to the glacier, the Collins Glacier here, until farther south in Valle Grande. Now the next expedition we are going to sampling this southern part of the island. This is the same study more or less that Jens Boy and group of Germany people has been working on. And probably we have to share some results for the same point, sampling point. So we also sampling Korea, Playa and Korean station. This is a Korean station, Kim Jong Korean station is very close to here. And we sampling 2 soil pits here in order to see a little bit more about the chemical point of view, characterization, features. And the idea is that this Korean Playa and Korea's sampling has a lot of, we were informed that they have a lot of iron content. Also here you find the Artoski Police Station. We are going to compare with the sampling that we did long before behind this project that we are going to present here. So general things, soil classification according to the USDA, key soil taxonomy. In general, the soil has already Kelly told us, is very loamy skeletal soils, I mean very coarse materials. And you have glaze soils, which is formed from cold factors like temperatures and very low organic matter. Empty soil, which are more or less soil which are very incipient. They don't still form layer or horizon there. Hilly soils, as already told you, and also inceptive soils. Inceptive soils we can find farther south in the island, and these soils have some development about the layers, and especially for the top layers. And also we have some ornithosols that I'm going to show you later. So the INACH project and NEXA project study the impact of freezing and thawing on soil organic matter construction after glacial retreat. I'm going to show you some slides about advancing our result, but some of the main results are still working on in the lab, and with this pandemic has been a little bit delayed, but still working. So our main hypothesis is that Antarctic soil might release a substantial quantity of CO2, as much higher as expected in the exposed polar environment. So the second point is that this could result in negative balance that hampers the accumulation of soil organic matter in recently disglaciated areas. And why is that? Why we put forward this hypothesis? First of all, we don't have much presence of vascular plant. The Sampsia antarctica and Colovanthus quitensis are the vascular plant that contain lignin. And second, the lignin is a recalcitrant component of the organic matter as a precursor of its formation, and is not present in the mosses and lichens. The third point is the Antarctic soil has been reported that great extractable amount of iron, chemical iron, that can protect soil organic matter against decomposition. So physophile microorganisms, which are phycrophiles, I don't know exactly how you pronounce that, but these microorganisms are very well adapted to the cold weather conditions, and they are abundant in these Antarctic soils. For example, iron-reducing bacteria might dissolve organic materials complex with organic metals complex with organic matter. And these bacteria are abundant in those soils, which are Antarctic ones. So I'm going to present the objective of our research here. Test first whether incipient clay formation or organomineral complex are the main soil organic matter stabilization agents involved in the soil organic carbon sequestration in maritime Antarctic soils. Second, establish how strong is the relationship between in situ CO2 effluxes and stabilizing agents such as clay and silt, as Kelly showed us before, or organomineral complexes. We are assuming that the context of the climate is very similar and very short transect of the island. So reveal, and the third one is reveal the biochemical composition of soil organic matter of maritime Antarctic soils. So we have here some pictures about the profiles, Collins Bajo, which is very close to the Collins Glacier here. You have a carpet of mosses, and this is the soil soil that we were 50 centimeters deep after you end up with a rock here, but most of this soil is made of cryoturbation. So you have at least 2 or 3 incipient horizon formation here. In Lago Uruguay, which is the farther south, you find more cryoturbation signs, and you end up with the 20 and 40, maximum 40 centimeter deep soil. And the parent materials is also volcanic, I mean basaltic, andesitic behind the soil, under the soil. Valle Grande is as farther south, close to the Escudero station. And you can see this is the same process is cryoturbation, which is the mixing of soil by the melting ice and water running through the profiles. So this is the Ardley ornithosols. Ardley ornithosols is island which are full of, I mean, it's very specialized, very protected area, sorry, and where the penguins rockeries are very common and other species. And the birds has a lot of influence on the soil formation. As you can see here, the carpet of lichens and mosses, and the penguins are living everywhere, nesting here. So the Korean Kim Jong-Barton Peninsula Station is shown here, and you can see that the soil is quite iron oxide presence. Here also you can have a layer of iron hard pant that can tell us that this soil effectively it has a lot of iron content. So some results. You can see the soil organic carbon component of this transit from close to the glacier and to Playa Korea in Barton Peninsula, also the ornithosols. And you can see that the soil organic carbon formation is very, very low, but except in ornithosol, which you might expect a lot of organic matter there. Also for nitrogen, the C2N ratio of those components, I mean carbon divided by soil nitrogen, is quite low. It's indication of rather readable, decomposable organic matter. It's not much very recalcitrant organic matter. Normally when you have a very low C2N ratio, you don't have a sort of immobilization, nitrogen immobilization in the soil, and mineralization, and also very labile material for decomposition. Soil pH is here, or is going to 5 to 8, which is normal. But the striking point I want to stress here is the iron extractable by diatomite. Diatomite extractable iron is the total iron content in the soil, and you can see that the Korea Playa has a lot of iron in there, and also the average of this 6 point that I show you, the 6 point of our study is about 1.87. So compare this iron content with the volcanic soil, which is reported high iron content in those soils is 0.93, which is quite low compared with the other one. Is 2 times higher in Antarctic soil compared with the continental volcanic soils. So why so high iron content in ornithosols? First of all, Nicol in 2010 estimated that the Southern Ocean krill population, krill is a crustacean, could contain about 24% of the total iron in the surface water, acting as an iron reservoir, far higher than background seawater concentration. Let's put this in context. You have this iron phytoplankton productivity. Iron is very necessary for the phytoplankton productivity because it's used as an energy cofactor for producing energy, and then for their productivity. And also the krill crustacean, which is living in the ocean, eat or consume this phytoplankton, rich in iron. And krill even don't need it so much iron, but they excrete it. And also young penguins in the rockeries can eat the krill iron rich, and also deposit the faces, rich faces on the Arley ornithosol. That's why it should be one explanation that we need to explore. But it seems that it's true that you have a lot of penguin rockeries with faces, a lot of iron on that. And this is the reason why the Arley ornithosols are very rich in iron content. So going back a little bit to the microorganism, we don't have the data yet for our microorganism in Fildes Peninsula, but this is a picture from Artoski Polish Station that we were studying before. And we made some analysis, phospholipid analysis, which was already published. And this analysis tell us the structure of the community, the microorganism community. Also, we have been done also genetic characterization. This is part of our characterization. And you can see that the Geobacter bacteria, which is the iron-reducing bacteria, is very, very large in basaltic Antarctic Arctosky, which is very close to the soil that we have been studying in the Fildes Peninsula. And the striking point is that you can see that the abundance of Geobacter here compared with the total amount of iron is very well correlated. Also for iron oxide, iron 3, and also soluble iron in the soil, which is iron 2. And this is a very interesting result because seems to be that iron reducing bacteria are very important in this Fildes Peninsula soils too. So some comments about the table 1 and figure 1. First of all, soil organic carbon and soil nitrogen are very low, except ornithosols. C2N ratio very low, indicate labile, readily decomposable soil organic matter materials, and high amount of iron, 1.87, twice the amount of found in continental soil like volcanic soils, and psychrophile microorganisms abundant in this Arctic environment. And these iron-reducing bacteria might dissolve organic minerals, components complex with organic matter, metals, complex with organic matter, releasing former protective soil organic matter now available for decomposition. This is more or less our schedule for make some inferences here. The inference is if anaerobic microorganisms are the leading agents for the release of soil organic carbon from organomineral complex, it would be expected that this organomineral complex can explain the stabilization of soil organic carbon. We were talking about this pH before. We were talking about the clay content and as agent stabilization agents for soil organic carbon, and we need to test that. So going back to our original question, whether organic mineral complex can explain the stabilization, you have here the relationship in the y-axis soil organic carbon and nitrogen are very well related, and from those results, we might plot the soil organic carbon content related to iron extractable with pyrophosphate. Pyrophosphate is an extractable reagent for organomineral complex that you have in the soil. It's a normal use for dilution, isotopic dilution also, for studying isotope with the different pools of iron on those soils. And you can see that there is a very nice correlation, almost more than 0.9 square, the coefficient determination. And also for nitrogen, you find this correlation. But if you plot it with the silt and clay content, which silt is also possible to gather some or retain some organic matter, and you see that the relationship is not very good, it's worse than the other ones, and might explain a lot about the main explanatory agent for soil organic carbon sequestration. We also had the opportunity to measure the CO2 in situ with the static chambers, where about 2 Celsius degrees in Antarctic soils. Here you have the CO2 release from the soil from 500 almost to 3,000 ppm in different soils. But also the important point to say here that this ornithosol, which is a lot of CO2 carbon content, 12% of carbon, can release almost the same amount as the other soils. Also, Playa Corea, ¿no es cierto? Playa Corea here is also with a very low organic matter content, but it's almost the same range of CO2 production here. And why is that? Well, precisely, when we correlate the CO2 from those sampling points, we end up with this plot with inverse correlation between iron link to organic matter linked to iron pyrophosphate and CO2. And you see that also the very nice correlation here, and both not so well as well the other ones, and for the clay and silt. So comment to these figures is that the organomineral complex can better explain the variability than incipient clay formation. Therefore, the destabilization of organic mineral complexes by psychrophile microorganisms like iron-reducing bacteria can play a key role in the release of soil organic matter decomposition like CO2 in Antarctic soils. So what about the quality of soil organic matter, which is also other possibility to have this early release of CO2 or easily release of CO2 in those soils? Well, we use attenuate total reflectance Fourier transformation infrared spectroscopy, and we select 3 bands here. And this band has to deal with the stretching vibration link or bounds for liable organic matter, OH and NH, and also this range here for amides and aromatics compound, also carboxylate compound, and conjugate ketones, which are in the middle of the spectrum. And in the extreme of the spectrum is the cellulose-like compound organomineral complex. And here you can see what is the spectra. We can see here the ornithosols, as we expected before, has a lot of iron oxide in the soil profile. Also, we very interesting result that the aluminium bound to silt forming crystalline clay are very high also for ornithosol, which is very high in clay content, compared with the Korean playa close to the Kim Jong station, which is almost with a weak peak here as on the second layer in the other playa Korea. So this diagram tells us that the same things that the organic materials in ornithosols are quite important, but these materials is also bound to iron and aluminium. That I don't tell you more about the aluminium, but most focus on iron in these ornithosols. So we also did a thermogravimetric analysis, which tell us the functional groups of these soils. Here is the diagram of heat flow. It means the energy you use per gram per Celsius degrees is a rate. Use it. Here you have the absolute weight when you increase in the temperature in ramp temperature from 0 to almost 600 degrees, you might find this loss of carbon content in your samples. And here these interesting things that the weight per milligram of gram and Celsius degree. This is the rate. As you can see here that this area is unprotected label organic matter in ornithosols. And this insectisol is Playa Korea. And this ornithosols is in the islands. And you can see that there's a lot of organic matter label one. But once the organic matter is released, I mean the Pond fraction particulate organic matter, which is very not really bound to the clay minerals, you end up with this almost the same amount as in Playa Korea, which tell us that there is a lot of complex of organic matter in those salts too. So the gravimetric thermogravimetric machine in our lab, in BioRen lab, which is a platform for big equipment in our university you can use. And this is nice things because this thermogravity machine is coupled to GC, and the gas chromatography has a library, complete library for to search for organic compounds or functional groups. And you can see here the functional groups, the abundance of functional groups according to the retention time of the columns in the GC. First of all, close to the 1 or 2 minutes, you get these CO2 gases, also ketones, and furans probably are made of because of the pyrolysis. The heating material here is a subproduct probably, but you have for certain security here the sugars and phenols at the end. But you can see that the Korean playa, this is the soil organic matter from Korea playa, and also for lichens. We compare both, and you can see that there's parallel declining of the materials that we found there. But the most important point is to stress here is that those materials has low phenols or aromatic compounds. So getting a little bit to the back to the conclusion in order to save time here. First of all, the Antarctic soil efflux were closely related to the organomineral complex rather than the amount of soil organic matter. Most organic matter is bound to iron content. That's why you don't find any correlation. As also Kelly showed us, there's no correlation between soil organic matter and the compositions. Well, I showed that, but Kelly showed something similar. And there is not a relationship with the soil organic matter and other type of soil properties. But the functional groups here, soil organic matter, were low in aromatic compounds, which are a labeled material for decomposition. And parallel, the functional group analyzed by for lichens also. So the freezing and thawing in summertime seems to be play a key role in the redox fluctuation and soil temperature for decomposition. Now we are studying the redox fluctuation system there in order to collect the samples and see in situ what is the fluctuation during this summertime thawing and freezing and thawing cycles. And also we has already told you, we are analyzing the microorganism across the soil profile along the island. Okay, I want to thank you very much, to thank the group here, lab group, which has been working very hard. Thank you for all your contribution, hardworking people and deep disposition. And I would like to thank INACH and Fondesit, Nexer, Universidad de la Frontera, and our lab. Thank you very much for your attention.
Thank you so much, Dr. Matus, for your wonderful presentation. Now it's time for the questions from the audience, so everyone can feel free to ask. All questions can be to Dr. Wilhelm or Dr. Matus, and they're going to be moderated by Dr. Matus.
Okay. Thank you very much, Javiera. Are there any questions in the chat, or in the chat there are some?
Yes, in the chat are some questions.
From Francisco Matus. This is thank you very much. This is in Spanish. I want to translate it. Thank you very much, Kelly, for your interesting presentation. I have some question for you. It is possible to make a relationship in having the soil not permafrost or permanent permafrost with the content of carbon in the soil? This is the first question. If you have some relationship between permafrost that you found and soil organic carbon in those soils. Or it's better you can answer this first and then I go to the second one.
Sure, I can answer. For the most part, I would say for our study area, at least, there's no relationship between carbon and those active layer depths, simply because the active layer depths are so extreme that it doesn't matter what's at the surface. Additionally, we did a lot of analysis of temperatures in bedrock that had no carbon whatsoever at the surface or nearby. So, and the bedrock had the same results as what we had in soils.
Okay. Yeah, okay. Francisco, are you okay with this answer? Can you hear me? Okay. The second question is that according to your plot that you show clay content and distance from the glacier, did you find any correlation between clay formation or clay amount and distance from the glacier front?
Yes, that's a little bit of a complex question. In some regions we did, such as in Sierra Point, where the slope was pretty consistent. We did find some correlation. However, there were many regions where the correlation was compounded by many other factors. Like in Palmer Station, there was a lot of intense water movement outside of the glacier. And so we had a lot of sands building up far away from the glacier where you wouldn't expect them based on normal water flow dynamics. Additionally, at Sierra Point, there were a lot of terraces and benches elevationally. And so those acted as areas, small retention ponds. And so we would have silts building up in those terraces regardless of their distance away from the glaciers.
Okay, okay. Yes, and the third one is, oh, let me see here. There is any relationship between the silt formation and glacier distance or relationship also more or less is the same question. Silt content compared with the distance of the glacier. I think it's almost the same question. Huh?
Yeah, my answer, I guess, would be pretty similar in that it really depends a lot on topography and geomorphic features for that too. So there is a correlation, but it's weak and complicated by factors outside of just distance from the glacier.
Mm-hmm. Okay. We have another question from Ignacio Joffre. For you too, Kelly. I would like to ask you, based on your experience, what is the importance of microorganisms in the formation of the soil in the Antarctic region due to the extreme condition of temperature? They are accelerating the formation of the soil. They are playing a key role in the progressive formation of the soil. This is a question. And what do you think will be result of this long-term phenomenon in Antarctic soil relationship with the carbon sequestration?
To the answer to the first part, yes, the organisms have a very strong effect on soil formation because they are producing organic matter and organic carbon that are going to remain in the soils for a long time. And that organic matter is going to play a role in things like retaining water for longer in certain regions. So if you have a very sandy soil, then water is not going to be retained very easily. It's going to infiltrate very quickly. But if you have a lot of organic matter at the surface that's part of those soils, the water can stay closer to the surface, potentially allowing for more organisms or plants or anything else growing at the surface to remain. And so you have an accelerated effect. Additionally, the accumulation of organic matter in our soils are going to have some pretty significant long-term effects in the development of these soils, because organic matter plays a role in helping to bind together particles into different soil structures, which make them more stable, more resistant to things like erosion and infiltration loss. And so the more organic matter that you have, the more stable your soils are going to become. And the more stable your soils are, the better the development horizontally your soils are going to have.
Okay, thank you. There is another questions here. Let me see, Carolina Merino. Do you evaluate the effect of one on bird near and far from the glacier? Because in areas far from the glacier, the amount of nitrogen and other nutrients in our ornithogenic soil are affected by leaching and lateral movements that produce natural fertilization of the soil. What happens with this nutrient provided by the birds in the vicinity of the glacier in permafrost areas? How they are incorporated or distributed in the soil? Well, those are for both of us.
Sure.
Kelly.
Yeah, no, that's a great question, and something that we at first were concerned about having secondary effects from the penguins on soils that we were assessing as neutral or acidic. The way we did that was to actually organize our soils based on phosphorus content, because the penguin guano has just a extremely large phosphorus amount that is 10 to 100 times more than a normal soil. And so it was very easy to separate a penguin affected soil, whether it was directly affected or affected by lateral movement of those that guano, just based on phosphorus content. Mm-hmm. And so we called an ornithogenic soil a soil that displayed large amounts of phosphorus in it. Mm-hmm. And so that's kind of how we separated them. It wasn't based on whether or not we saw penguins or anything like that. It was a chemical separation. And we recognize that probably a lot of our soils, especially the ones that are marked as acidic, probably do have a little bit of effect from the penguins, but it's impossible to be able to fully separate the ornithogenic soils, the penguin affected soils, from the non-affected ones. They all have a little bit of effect, and that's part of the acidification process. So we tried to keep that in mind as much as possible.
Okay, thank you very much for your answer. So we have also some questions. Jens Boy from Germany say, I'm sorry I have to go. Was very interesting talk. Thank you very much. And the other one is Francisco Nájera. Thanks very much for your answer. We also found great variability in our data that will be really interesting to follow. So this is a general comment. Are there any questions from the audience that you might find interesting to ask us? So
it seems like no one has more questions. So now it's time to close this conference. So I would like to ask Dr. Matus to give a few words, maybe a little speech as organizers of this delightful webinar, please.
Okay, thank you very much, Javiera. Well, I would like to thank all the participants. We were a little bit, I don't know how much people was in the beginning, but now 16 participants. And I would like to thank you, all of you, and also Kelly for a nice presentation. And well, I hope that we can have a contact in the near future You have our contacts there. Any question or any requirement, you know where you find us. Thank you very much. And so for the next time.
Thank you, Dr. Matus, and all your organizing team for this instance that was surely very fruitful for all of us, covering contingent topics related to carbon cycle and maritime Antarctic soils. So we would like to thank all our audience for staying and being part of this webinar. That being said, see you at the next occasion. Have a wonderful day. Goodbye.
Okay, see you. Bye-bye.